Philip,
and
Emad,
Bio
Lab.
World. Alright, so today we're doing experiments testing enzymes in their optimal temperature and their optimal pH levels.
So what happens when you leave an apple? You cut an apple open and you leave it lying outside. Doesn't it turn brown? That's because the catechol oxidase in the plant cells produce
benzoquinone.
big word. Produce that to protect. It's toxic to bacterial cells, so it protects the inside and it protects the plant or the fruit.
So, as you know, we are talking about the optimal temperature and optimal pH for enzymes. And we are testing the catechol oxidase enzyme that is found in plant cells, in fruit, which reacts with catechol and oxygen when it is exposed, and produces the product benzoquinone, which is an antimicrobial agent, which protects the fruit. So here we have the first test tube, okay, where we put in 1 milliliter of catechol, no water, and 1 milliliter of catechol oxidase enzyme.
Thanks, folks. Take some notes.
Notice the color. That color comes from the benzoquinone. Okay, this dark brown color comes from the benzoquinone, which is the product. You see the catechol as the oxygen reacted with the catechol, and the enzyme came in and produced that. Here's the second test tube, where we have 1 milliliter of water and 1 milliliter of catechol oxidase. Notice the color. It didn't react, and there's no benzoquinone produced. This is because there was no catechol. You only have two parts to your reaction, to your equation. Without the catechol, there's no catechol oxidase reaction. And here we have final test tube.
1 milliliter of catechol and 1 milliliter of water. Now we didn't add catechol oxidase in this test tube, and as you can see, there is no brown color because since the enzyme we didn't add the enzyme, there is no what is it called?
Benzoquinone
produced.
So, if this was at a higher temperature, then you may have seen the benzoquinone reacting with the catechol and the oxidase.
Because you would have gotten more energy.
What an enzyme does is lowers the level of activation energy. So your chemical reaction requires a certain level of energy. Okay, and when you have you have your starting reactant here, you have a higher level of energy activation, and then you get your product down here. Okay, so it looked like a curved graph like this. And what an enzyme does is it takes this activation of energy and it lowers it so that the reaction can take place with less energy. Okay, so as we were talking about these before, I only have 2 of the test tubes right now because the third test tube that we had, only the catechol and the water, we are continuing to oxidize, or to incubate, to see if we can get any chemical reactions from it. This test tube number 1, as you remember, which produced the benzoquinone, is our positive control. So any reaction would have a color like this. Okay, maybe a little less shade, a little lighter of a shade if it's less of a reaction or less produced. And this is our negative control, along with the milliliters of water and milliliter of catechol, the one that is being incubated as we speak. Welcome back to Emad and Phillips
Lab World.
Okay, so here we have our test result: 37 degrees, 1 milliliter of water, or 1 milliliter of catechol, 1 milliliter of water, and 1 milliliter of catechol oxidase enzyme.
The same thing is added to test tube 2 and 3 respectively.
2, 3. Okay, we had the same solution, but we tested this one at 37 degrees,
this one at 60, and this one at 80.
So as we compare this to our positive test result, Mr. Philip, if you could just, there we go. Here we have our positive test result from our first run. Notice the color; they're very similar. meaning that test tube number 1 had a very good positive test result. Here, we compare positive control with number 2. I'll take test tube number 3, or test tube number 2, sorry. Test tube number 2. Positive control. This is lighter, showing that this had less of a reaction. Okay.
And you notice there's precipitate at the bottom of the test tube. At higher temperatures, the cellular debris tends to coagulate, and that is what the white stuff at the bottom is.
Okay, because to get the potato extract that we use for the catechol oxidase enzyme, we took a potato and blended it. Mashed potatoes.
Yummy.
So, we see that this has been denatured by the heat, showing that it didn't have as much of a reaction. Okay. And now, compared to test tube number 3, positive control, test tube number 3. As we can see, precipitate at the bottom, and almost no brown. Very, very light, and it's hard to see, but there is very little brown benzoquinone. So we can see that at 80 degrees Celsius, the enzyme was greatly denatured.